Welcome to the Cannabis Cultivation and Science Podcast. I'm your host, Tad Hussey of Kiss Organics. This is the podcast where we discuss the cutting edge of growing from a science-based perspective and draw on top experts from around the industry to share their wisdom and knowledge. My guest this week is Colin Bell. Colin is most well known in the industry as the co-inventor of Mammoth P. He completed his Ph.D. in Soil Microbial Ecology in 2009. As a research scientist at Colorado State University, Colin published dozens of peer-reviewed publications that were focused on elucidating microbial-mediated processes that enhance plant growth. Colin's academic research includes long-term multidisciplinary studies in which biological, chemical, and physical characteristics of soils were measured to elucidate soil microbial and plant responses to climate variability. He currently works for Miicrobial Mass, a plant amendment product comprised of several unique species of beneficial bacteria shown to promote plant vigor, increase plant biomass, and enhance microbial life in and around the root zone. Now on to the show. Hey, Colin, thanks for coming on the show today.
Tad, great to see you. It's been quite a while and happy to be here.
Yeah, I'm glad we got to reconnect, and I wanted to just start off. Why don't you tell us what's been going on with you, a little bit about yourself?
Yeah. So for those of you that don't know me, my name is Colin Bell. I'm actually a scientist in my background training. I have a Ph.D. in soil microbiology. I studied at Texas Tech University. From there, I moved over to Fort Collins, Colorado, where I worked at the Natural Resource Ecology Center at Colorado State University. And I worked for years, and all my academic and research background is focused on understanding the importance of plant microbe interactions. Did a lot of studies, published a lot of papers in both natural and agriculture systems, and just basically got a really sound fundamental understanding of how microbes work with plants to support plant growth and in these different environments, including precision agriculture systems and indoor growing.
Awesome. You also were the founder or creator of Mammoth P. So I want to mention that. That's pretty cool. Yeah. And now you're currently working over at Miicrobial Mass. Is that correct?
Yeah, that's right. So, I was the co-founder of Mammoth Microbes, the inventor of that technology. And a lot of people probably know me because of that. And it's been a really amazing journey. And I learned a lot doing that. Ultimately, and it was at the very end of last year, in October last year, I made the choice to leave the organization for a lot of reasons. One, it kind of got to the size where I thought it was going a different direction that I didn't necessarily want to go. And it wasn't in the vision, but when you get to a larger size organization and bring people in and outsiders and investors, you start to lose control and maybe swerve away from the original vision and intention you had for the organization. So I wanted to follow my path. And I left. It took some time off, and I've done some fun stuff, and you and I were talking about some of those things. Ultimately, trying to follow my passion and trying to really enjoy my professional career to the fullest. And I enjoy that by working with people that I really enjoy working with, that are go-getters and that I can trust and have a lot of fun with. And along with that, I have to work with companies that I believe in. And not only from a mission and vision, but from a technology and innovation point of view. So it has to kind of fit my expertise, which is microbial and natural technologies for agriculture. So I'm very passionate about agriculture innovation and figuring out how to formulate and how to develop nature and nature solutions to fit into conventional agriculture management practices. And the Microbial Mass Technologies, MIM Horticulture is the name of the company, out of Canada, has done some very interesting things that intrigued me, not only from a team perspective, but from a technology perspective. And so I'm really happy to be helping them develop their business in the U.S.
Yeah, I was kind of blindsided because you and I had talked, and I was just asking you what you were up to, and you weren't doing anything. And the next thing I knew, I hear from another friend that you're working over at Miicrobial Mass. So I had to call up my buddy David Bernard Perrone, and he was kind of filling me in. And then Francois, who actually was the one who developed Miicrobial Mass, is another friend of mine who I haven't talked to in a little while, but he's an awesome researcher, and so I'm excited to see you guys working together. And before we dive into like the real meat of the podcast, tell me just a little bit about microbial mass, and what you find interesting about it as a microbial, biostimulant.
Yeah, thank you. And I'll just mention that, David and Francois are two of the reasons when I speak of team and people that I really want to work with and have fun working with, was part of the decision making for me joining this group. Great guys, a lot of fun. And I know you know them both very well.
Yeah.
And, I really geek out as a microbiologist and someone that really wants to apply microbiology in the market with technology and microbial technologies. And the approach that I took with Mammoth P was a quite different approach. This group took quite a different approach, which intrigued me. And it was something I've been trying to do or wanting to do with the Grossentia group when I was, an operational founder and, member of that particular team. And I saw this group being able to execute that well. So it's really fun. And the differences, I think, are precision, to be fair. There was some certain functionality that we nurtured in Mammoth P, targeting phosphorus cycling, but there was a lot of unknowns that were occurring in that particular formula as well. And as a microbial ecologist, soil ecologist, I embrace those unknowns, knowing that there's not everything we're ever going to learn about plants and microbial function, but believing that a lot of those things could enhance plant success. And there could be a lot of functional redundancies that could help plants grow successfully across the growing season without us necessarily understanding what they all are. What I felt like the MIM Horticulture group did really well was work with super defined carriers, super defined formula, and really honing in on the functionality of the microbes they were using to not only present a very specific microbe with a very specific function for the industry, but it eliminates a lot of the unknowns. And it made me think of something that was pretty common in the industry now, at least from the salt and the hydro growers, was this aspect of clean. And a lot of the clean speak around growing plants now and keeping it as clean, as sterile as possible. It's not necessarily something I completely agree with, but I understand it's the paradigm, and a lot of people want to start focusing on that aspect of growing. And I felt like if there was a way that we could incorporate microbes into a clean grow environment, it would help the grower. But we had to figure out how to do that well. And I feel like this group has done that with this particular formula. It's the first clean microbial solution where everything's very well defined, and they're using dormant bacteria to deliver to the plants. And so it works through the irrigation fertigation system completely virtually sterile because of the dormancy of the microbes, and the microbes actually activate when they get to the plant roots to stimulate plant growth across the growing season. The Mass product in particular has done a couple things. They have a couple microbes that they've quantified really well in the laboratory and in grow environments to increase the mass of early stage rooting. And then 3 of the other microbes in this particular product are focused on later stage phosphorus cycling. And so it's a really interesting 1-2 punch, maximizing the plant's ability to establish early on and then to uptake nutrients later on in the growth cycle to maximize their ability to stack and yield.
So you don't have the same issues with biofilm buildup, I would assume, if you're looking at dormant bacteria. And then this product was developed, I believe, or discovered out of David's grow when he was at Whistler Medical on the cannabis side. So the product was actually derived, and correct me if I'm wrong here, but from that sort of relationship, coevolution of these particular bacteria with cannabis plants.
Yeah, how I understand it, the product wasn't developed, but the microbes were identified in that environment. Yeah, and so they were identified, then they were selected, and then scaled up, and that's where the product development formulation process took over where they utilized the microbes from the cannabis grow to create a clean formula to carry them in to make it very compatible, not only for living soil growers, but also for clean, salt, hydroponic growers.
Yeah, I think you bring up an important point. There's a big difference between clean and sterile. You can have microbial interactions going on in your soil and not be sterile, but still have a clean room. And, we're not throwing out all the IPM processes around keeping your area clean. So I just want to highlight that. And then, I'm kind of going off topic here a little bit, but you and I just both attended a webinar by Dr. Punja talking about hop latent viroid. And one of the things that he pointed out around it was that it damages root mass in clones. He had a lot of interesting points around it, in terms of most plants being asymptomatic and just the prevalence of this viroid in the plants that we are currently cultivating. But I wonder if we could have a reduction in symptoms if we can get some microbial products that potentially could increase root mass to offset some of the damage that we're seeing from a viroid.
I think conceptually that's exactly right. What we heard in that particular presentation, which was very well done, was that in a lot of cases it silences genes in the plant among a host of other things that the viroid does to the plant to help kind of deter its maximum success, diminishes root growth, diminishes trichome production, diminishes overall yield, diminishes stem stability. But the gene silencing I thought was pretty interesting also, because that's going to silence a lot of the resistance genes in these plants. Mm-hmm. And so the idea is maximizing the plant health to its full potential. And there's a strong argument that if you have the beneficial microbes occupying the rhizosphere and root zone, it's going to maximize the plant's ability to fend off particular pathogens. The other piece of that was fascinating. The viroid, hops latent viroid, actually makes plants more susceptible to bacterial infections. And so you can almost offset that with beneficials because there's a probability gain. Life is a probability, and if you can occupy space with beneficials, it's going to be harder for pathogens to come in and affect your plant.
Yeah. Well, let's getting back to the outline that I had for this conversation today. One of the things that I thought would be really useful to ask you is, how do we grow microbes? This is something that you've had a lot of experience on, especially when you were developing your product. But for the home cultivator or a commercial grower, they don't have a lab. They're probably getting things off the shelf as food sources and trying to use microbes like maybe lactobacillus or maybe extend a microbial product they already have. How would someone begin to start, I don't know, doing this process?
It's really simple, and it can be a lot of fun. I recommend it for everybody. And I used to teach at the university when I was a student. I would teach labs, and we would culture microbes from the environment. And so that's a really fun way to start if you're thinking about growing microbes. And I've had people take a sterile swab. You can take a sterile Q-tip, and the growing microbes I'm going to start is not as complicated as keeping them pristine, meaning growing the microbes you want to grow, and preventing from contamination. We used the word sterile a minute ago, and that's something that comes up sometimes in cultivation facilities. But in general, it's a misnomer. There's no chance that sterility exists in the environment. It's a probability. But sterility just doesn't happen. Yeah, it might be able to happen for a short period of time, but you can grow. There's a lot of different food sources that you can make or you can actually commercially buy, and they're pretty cheap to grow microbes. And so just like you and I, microbes are heterotrophs. They eat food and they respire carbon and they take in oxygen. And so you can just create any kind of food source and you will grow microbes. The interesting thing about growing microbes is some favor some food sources over the others.
Mm-hmm.
And so there's a lot of different media you can use to maximize the success of growing certain strains. There's Bibles of microbiology, so to speak. It's a methods of microbiology where they have thousands of different microbial medias and they have the actual formulas for them. And so it's a really interesting book you can reference. You can probably buy online. You can definitely pick up at a library or a university library for sure. And just look at all these different ingredients. There's typically sugar sources and there's typically protein sources. And there can be a liquid solution or it can be in a solidified solution where you have an agar gel from polysaccharide to help solidify that. And streak microbes or streak something that you've collected from the environment on a plate or just put it in a broth and microbes will grow. You can actually leave these plates out just exposed for minutes, close them up, and you can grow microbes from the air and you'll see growth in less than a week easily. So it's very simple to grow microbes. Understanding the microbes that you want to grow and how to optimize that growth becomes a lot more sophisticated. And with all kind of microbial production, the growing of microbes is typically not the crux. It's the maintaining the purity of that microbial culture is what becomes very challenging. And so the aseptic transferrin technique, which is your sterile technique of transferring from one place to another, your microbe, becomes most of the risk when you're growing microbes. That's why it's really hard to have a consistent microbial formula over time. And I imagine most people, if they tested their lactobacillus or any of their concoctions that we make, which is cool and I love it, and including microbial teas, I think we'll find very different microbial assemblages in those over time from batch to batch, seasonally. There's from the water source, there's so many different sources that can affect that microbial community and the structure of that over time.
Yeah, so I came into this industry through the aerated compost tea world. And what you're saying really holds true for my experience too. And that was that, every batch is going to be different. We're not identifying these particular strains or species, but by having sufficient levels of oxygen and selecting an aerobic environment and using a source of material, in this case compost, that we know to be fairly free of pathogens, we're hopefully like tilting the scales in the direction of more beneficial microorganisms, or at the very least, what I think, and when I talk about compost tea, people make claims that I think are just crazy around it. Like for me, the idea of disease suppression and some of these other things may occur, but like you said, every batch is going to be, or like I said, I guess every batch is going to be different, and we can't get replication consistently. So it'd be really hard to make any conclusions, or even determine any trends over time. But what I think is interesting about it is, can we get nutrients, functional like nutrient cycling, like in a tea, that we can apply to the soil that would allow us to increase the, that microbial loop, that nutrient cycling going on some level through sort of this like shotgun approach.
Totally. And I think the answer is absolutely positively yes. And it's because of the way microbes work. And what I would say is a really practical goal for a tea or having just a mixed consortia of microbes is having a critical mass, a number, a count of threshold biomass that gets to a certain level where it can function at a very high level. And it doesn't necessarily matter who's in there in every case. If you're trying to get a general nutrient cycling function out of a microbial group, you want to make sure there are no pathogens. And you can make the assumption that there's enough functional redundancy that you're going to have microbes that under different conditions are going to step up and perform the function that maybe another microbial group that performs the same function can't under a certain environment. And you see that a lot in nature. I don't see it any different than in natural systems. And I used to do a lot of work in desert ecosystems, and we used to talk a lot about functional redundancy because the environment was so extreme that you'd find different microbial groups that you could test them, and they would do the same thing functionally, but they would have very different environmental thresholds. And some would work much better in drier conditions or in warmer conditions, and some would work better in colder conditions or under pH stress. And so having the functional redundancy, I think, is a plus. And the real value in those cases is making sure you have a certain concentration so you can maximize the functionality. And so there's going to be this asymptote, if you want me to draw a line here with my hand, where the lower the concentration, you're going to get lower functionality. Then at some point, it's just going to reach this critical threshold where you're maximizing your functionality as a function of the biomass or the concentration of microbes. And reaching that without any potential pathogens would be, I think, the target that you're trying to go for in a tea or any kind of microbial concoction that's generally defined.
Yeah, so when you say functional redundancy, essentially what you're saying is, just to reiterate, in a desert system, for example, let's say cactus needs phosphorus, and there is a particular strain of bacteria that's very good at cycling phosphorus when it is very cold out. And then it heats up, that bacteria is potentially going to go dormant, die off. Well, the plant still needs phosphorus. Well, another microbe that maybe doesn't function at cold temperatures but functions quite well at hot temperatures, say over 100 degrees Fahrenheit, is going to reproduce and cycle nutrients and provide phosphorus for that plant under those conditions. And so they essentially can occupy the same ecological niche for the plant, which is phosphorus cycling, yet coexist and not really compete with each other because they're functioning at different variables. And like you said, it could be pH, it could be water, it could be heat, it could be cold. Does that kind of encapsulate that?
That's exactly how it works in nature, and that's exactly how it would work in farming or in precision agriculture, indoor growing. pH is the example that I think is the most prevalent environmental factor affecting microbial success. There's a salt concentration with nutrients and all the just conventional management practices, and pH is a big part of that also. pH actually is shown to predicate microbial diversity and community composition more than any other factor, being the strongest factor to manipulate microbial community structure across the world. And so you look at different conditions, or different soil types, whatever, out of all the factors there might be, temperature, water, pH, others, pH is the first determining factor. And I think it's interesting for indoor growing because you can have pH shifts. You try to avoid that, but that happens. And so with functional redundancy, you have microbes that are able to persist and function through those shifts and kind of overall bridge a functional gap where a single microbe might work really well at a certain pH, let's say 7, but it might be very stressed at a pH of 5. And so that would not be the best microbe in that particular environment. And having another backup microbe that worked well across the range of that particular environmental threshold, pH, would serve the plant better.
That's really interesting. I think it's important for people to keep in mind that we tend to think in like black and white, but the reality of the complexity of the rhizosphere is that like the soil may have a pH that we're getting, but like within that soil, there's all these little micro pockets where the pH might be a little bit different, where hydrology might be a little bit different. And that's where these little ecological niches occur. And it's hard for me to wrap my brain around because it's so small and it's constantly changing. Like this idea that the microbes in your soil are always changing, I think is really, really important, because when we start getting into these like general thoughts of like fungal to bacteria ratio or, overall biomass, if my soil is always changing, how can we use those metrics to really make informed decisions just due to the variability and fluctuation of it?
That's a great question. I'm going to add to that in soils, if you compare the different types of substrates, let's say that plants might grow in agriculture, indoor agriculture, it can go from soil all the way to soil as media. Then on the far extreme, let's just say rock wool. And so there's different buffering capacities across those substrates that vary substantially. If you think about the rock wool scenario as the most extreme other example from soil, those environments are going to change dramatically with every watering event, because as the ions coming through and it kind of drains through, it doesn't have a huge buffering capacity. And so you really want to be diligent on pHing the solution before you water your plant, because you can stress that. But microbes work really well in those environments. On the whole other extreme, I think about soil where it's more buffered and in the rhizosphere. And you think about microbiology, and scale is a really interesting topic. Microbes are just micrometers, which is such a small, such a small size that obviously it's microscopic. You can't see it with the naked eye. Mm-hmm. But if you saw it along the tiniest root hair, it would just be a speck, even from a magnification. And so the environments that get created along the root zone or the rhizosphere are highly buffered. And so I think that you're going to have an ebb and flow of different populations, but there's also, I think about it as a probability game, where if there's successful microbes in that environment, they're going to swell and shrink depending on certain circumstances as far as their overall population or growth, but they're probably going to remain there. Mm-hmm. And they might not completely turn over to a completely different group. You might have some additions coming in, but I don't think that the turnover is as extreme as much as the ebbing and swelling would be. So it's kind of a probability game.
Yeah, and then the other thing that further complicates it is the life cycles of these microbes. The generations of them are so fast. So just within a given flowering cycle, we have co-evolution happening on a microscopic scale. And so, and I think I've mentioned this in previous podcasts, but I talked to my good friend Nate Gibbs over at Gold Leaf Gardens, and they've had their soil in their beds for over 8 years now, and that's his intellectual property at this point. That's what gives him something different than everyone else on the market, because the microbes in that soil, I mean, we'll call it microbes, but the nutrient levels, the microbes, the pH, all of those things are unique to him. It's no longer the soil that he got from me 8 years ago. It's his own thing. And I think that's really interesting.
Yeah, it's totally cool. I think the visual I would use to frame how microbes exist in any environment in the rhizosphere and soil is how I talk about it. As a community starts off, it's just a very low concentration or a certain single microbe. And they call that the lag phase of the microbial growth curve. And at some point there's typically a lot of food resource. Ultimately, that microbial group or that mass will get to a certain concentration, and they grow really quick, and they reproduce or clone out very quickly. They split, and they asexually just clone out, and in days they can really change their mass. But then they'll grow into an exponential growth phase where they'll just grow at a 10x rate in that environment very quickly. And at some point they'll level off where the food source equals the live death. And so the food source is sustaining that certain concentration of microbes. And at that longer growth phase, it's a life equals death. It's called stationary, where microbes are turning over, but they're reproducing at about the same rate they're dying. Mm-hmm. And so they're maintaining this certain mass. And only when a food source or some environmental conditions really disrupt that particular mass, you get this death phase going down where the concentration starts to decline rapidly. And so that stationary growth phase can persist for a very long period of time. And it's just indicative of the microbe being very happy in that environment and having enough of a food source and a long enough life cycle to persist and have a pretty predictable turnover, if that makes sense.
Yeah, and when you say food sources, that could be a variety of things. That could be exudates coming directly from the plant itself, out of the roots.
Totally.
It could be a variety of organic matter things, or fertilizer for that. For example, it could be a little bit of
Totally.
alfalfa meal, a little bit of feather or fishbone meal. It could be salts. Let's talk about mineral salts. How do they affect microbes?
Yeah, it's a great question. So, a lot of people think that mineral salts actually kill microbes. And I'm here to tell you that's not necessarily the truth. It all depended on time and concentration for even disinfectant solutions. I'll tell you what, I've tried to kill microbes before, or robust microbial formulas, and I've had a very hard time doing it, even with commercially available products. Hydrogen peroxide, chlorine solutions, and it comes down to the concentration of the solution and the time. And over time, those are the two factors that you have to dial in order to successfully kill microbes in general. In practice, I've never seen microbes really die off through just a fertigation practice. I've actually had a hard time again killing microbes through just recommended practices of most disinfectants that are recommended in our industry, cleaning lines and whatnot. It has to be a longer time and a high concentration to significantly disrupt microbes.
So the idea behind salts killing microbes is that they raise the soluble salt level in either the media or in the nutrient solution. So the EC of that to a level to where the microbes through osmotic shock die, essentially. So there's been this idea perpetuated in the industry that mineral salts kill microbes, but yet there's research out there that I've seen that shows that mineral salts in low doses actually feed microbes. They're a source for a food source.
Microbes consume the same nutrients that plants do. And the only other nutrient that they get, but not from the air, but from the substrate, is carbon. And so this is a pretty interesting evolutionary tool of plants to feed photosynthate priming into the soil or to the substrate to feed microbes the sugars that they take from the air. They consume some of the carbon to create their biomass, but they also shoot a lot of that sugar into the soil to feed microbes the carbon they need, so the microbes can actively help cycle nutrients to support their success. And I used to say this all the time, but microbes, all plants in nature, the natural ones, exist because of that microbial relationship they have in the soil, because the microbes actually help them take up nutrients. And if you're feeding soluble nutrients into a solution and microbes are there, they're absolutely going to take advantage of those just like the plants would. And so I wouldn't call it a competition, but there is a little bit of a cohabitation of utilizing the resources in that particular environment.
Yeah, I think that's just an important point that I wanted you to make, or to hear you say, because what we see from when we look at like competition vegetable growers, like giant pumpkin growers, tomatoes, things like that, they're doing a combination of organic inputs, microbial stimulants like mycorrhizal fungus, for example, and then levels of mineral salts, and that's where we see the largest yields. And I think, like, ultimately, if we want to talk about just growing the largest plant, it would be this hybrid method. Now, I'm an organic guy, and I could absolutely make the argument for why I choose not to use mineral salts. But I also want to be pragmatic about it and acknowledge that, if we're just talking about yield or plant growth, this hybrid method, I think, is really the way to go.
Absolutely, positively. I mean, that's what I've seen also for a couple different reasons. I think that having that extra catalyst, if you will, if you were going to call microbes a catalyst, and they do not only act as a catalyst, but they create other biomolecules that act as catalysts to help cycle nutrients, to help stimulate the plants to want to take up more nutrients. I've seen the rhizosphere with beneficial bacteria protect plants from infection, from other pathogens that might come in just from ecological niche theory by itself. These microbes can create different chemical signaling analogous to hormones in some cases that they'll stimulate plant growth, which is a very interesting thing. They obviously cycle nutrients, which can become sticky and not available to the plants, especially in soil. Phosphorus is a great example of that, where they can actually solubilize that phosphorus and make it available for the plant to uptake. And so there's a lot of things these microbes do to help plants across the growing season. I guess one more example is, and this isn't as much in soils as it would happen in probably cocoa and rock wool in some cases. And there's a lockout event that occurs in some cases if there's a flooding of ions and the plants are able to balance that out very well. And microbes do a really good job of balancing out plant nutrient uptake along the rhizosphere and root zone. Mm-hmm. And microbes can actually alleviate the potential for lockout events. And I think that's a really fascinating thing that a lot of growers probably experience just without understanding it. You can have these really micro lockout events through just feeding salts. The plants won't show for days and the plants can probably work out of it, but it's a staggering growth event that with microbes, the plants wouldn't feel that. And they could focus all their energy just into the upward growth of the plant instead of the plant being almost stunted. Although you couldn't measure that necessarily just by watching the plant grow as it's being fed pure salts across the growing season. And I've had growers that have used microbes that they were just feeding, rock wool is an example, salts, and they would add a microbial solution. And in 2 days, they would just see their plants open up and literally kind of, they would call it explode. And like I said, their buds just started really growing. And it took me a while to think through this, but I think the truth is that there's a lockout that's occurring that microbes completely get rid of and allow the plants just to focus on uptaking nutrients and allocating them without any kind of stutter.
So your theory or hypothesis around this is that even in a relatively sterile system, and we say sterile, but there's no grow that's actually sterile.
Right.
But in a system with less microbes that's not using soil, using a media like rock wool and mineral salts, there's still room for clean microbial products that could potentially increase yield or have other plant benefits.
Totally.
And
I've seen it.
I get it. I don't know that I fully understand the mechanism of action there. I'm thinking, okay, these microbes maybe could help potentially cycle nutrients. Then are they surviving or persisting in the rock wool, or are they converting nutrients into a form that addresses some other plant need? Are they occupying niches on the plant roots themselves? Like, what's really going on? Do we know?
I don't think we know completely. That's what's kind of interesting about it. I would say that in a rock wool environment, since you brought that up, the rhizosphere and roots create a very habitable environment. It's a sticky, nice carbon foundation, and it's exuding carbon into that rock wool. The plants are just doing that. So the microbes can live on the roots and be very happy, quite honestly, no matter what the substrate is. And you'll find even in soil, the microbial concentration gets greater the closer to the roots or on the roots. And so that's the hot spot that the microbes like to be in. And a lot of microbial science is still a little bit nascent. It's hard to measure all of the idiosyncrasies and interactions that occur.
Yeah.
But I think part of that turnover that you referenced earlier, where the microbes are going to die, they are going to lyse, these cells break open, and the constituents of those cells create a biofood source that stimulate the plants, and the plants are able to take that up also. There's a lot of simple proteins that the plants can take up that way. And ultimately, what I've seen is I think the plants just get more nourished, or they are able to get more resources with that extra catalyst somehow. Obviously, we understand nutrient cycling. There are some other mechanisms, biological mechanisms that are obviously occurring in a more synthetic environment and a more of a rock wool. substrate where the nutrients are being fed and we think that they're all soluble, so the plants seem to be taking them up.
Mm-hmm.
But I think plants in some cases are able to take up more nutrients, and so they're able to allocate more.
Hmm.
And one last detail that I think is fascinating, I've seen just numerous times in, say, a rock wool grow, and you can have a synthetic fertilizer and a synthetic fertilizer treatment with microbes, and the plants both look good, but they're actually not the same. And I've noticed in many cases the smell, the terpene profile, and even resin profiles, anecdotally and quantifiably, are much higher in a lot of the microbial treatments than just a plain synthetic. And you can even tell by the smell and going through. And I've smelled plants that looked similar, but one smelled like hay or cut grass, and one smelled more like a really interesting cannabis strain, and it was sticky. And the only difference was microbes. So I think that the microbes are not only able to deliver more resources to the plant, but they might also trigger some type of signaling that allows the plant to turn on some of these genes that produce more cannabinoids, maybe produce more terpenes, produce more resins.
Yeah, I mean, we know now that some of these microbes are actually going into the root tip itself, into the plant, and then back out again. It's pretty incredible, the stuff we're learning.
Yeah. Yeah.
So one thing I've been thinking about lately, because we were talking about sort of how how do we apply microbes, or how do we look at soils and diversity? And there's this is not a complete thought, so bear with me on this. The first part of this that I want to share that goes into this thought process is when I make compost tea, I'm going for nutrient cycling, right? Well, when I look at it under the microscope, if I brew for, let's say, more than 60 hours, at the end of the day, I don't have that level of diversity anymore. Pretty much what I'm down to is just like one morphology of bacteria and one morphology of typically of some sort of flagellate. It's like, and I give the analogy, like, what if I just took a zoo and put out all this food and opened up all the gates? At first there'd be this explosion of growth, and then you'd come back a week later and you just have like your lions and your rats, your apex predator and your best, survivalist, and they'll fluctuate. Yeah. So the bacteria will explode, and then all of a sudden, I'll come back 8 hours later and it'll just be all flagellates because they will have eaten all the bacteria, and then they no longer have food, so they die off, and then the bacteria explodes again, and you get this like roller coaster, but you've lost all your diversity. And so when I think about no-till, you can see where I'm going here, and this idea that my good friend Ben Higgins explained to me, talking about the disturbance hypothesis, which is this idea that when we have, new land that has, let's just say a volcano, lava forms this land. There's no life on it, right? We'll slowly start to see species get introduced, but the diversity on that, on that rock is minimal. Well, over time, that diversity increases, and we'll start to see grasslands, and then forests, and soil will obviously develop, and then eventually we'll end up in what we is known as like a climax ecological niche, something like old growth forest, and the diversity declines at that point. So somewhere in the middle there is where we have our maximum level of diversity. It's not at the end in the old growth forest. It's not at the beginning, where we just have bare rock. It's somewhere in the middle. And so this is one of the reasons, like in my brain, I think, okay, maybe a little bit of soil disturbance is okay. Maybe it's good. So when we go to re-amend our living soil beds, we're not breaking out a mechanical tiller and just destroying all soil structure, but we are mixing in nutrients for the next cycle. Is that really as harmful as like people might make it out to be? I think I've already kind of shared my opinion through my explanation, but what are your thoughts around it?
Yeah, well, what I'm kind of hearing is secession theory, which is a really fun idea. We used to do that in my ecology class. I've grown soil fungi for years. Yeah. And as a desert ecologist, fungi really dominate the function of those soils because of their not only the morphology but their physiology, and they're able to withstand. And you can see that at a microbial scale. You can see this on this forest scale that you're talking about, and this kind of peak of diversity before it starts to dwindle. I've always thought of agriculture as an inherently disturbed system.
Mm-hmm.
Like, I don't think there's any kind of stasis or succession where this natural environment slowly, the biology in that slowly evolves in succession. So you have this just climax type of community. It's not going to be that way. And so I think that in agriculture, it's a balance. I think it's whatever you're trying to accomplish is really important. I would say that fungi get more disturbed than bacteria just because of the structure that they form.
Mm-hmm.
And so if you have just in really intricate, involved, established fungal mats, and you break that apart, that's going to disturb the functionality of those fungi. That's not necessarily the case in our systems. Bacteria are a lot more forgiving through disturbance because their size allows them not to feel the disturbance, mm-hmm. like a larger organisms might be. You think about farmlands where the tilling versus no tilling has been argued, and I think that there's a practical balance where you'll see a lot of farms where they have strips of grass in between every other row, and that's to nurture the mycorrhizae and the fungi in the soil, where there's at least a presence of undisturbed fungi that can help influence the plant growth mm-hmm. Every year as they're planting out. So I think it's a really hard target to think you're not going to disturb soils or work with disturbed soils in any type of agriculture. And I think it's important to understand, if you're using biology, to understand how resilient that biology is that you're working with in that particular environment. So not all microbes would do well. If you're collecting, to go back to your example, if you're working with tea and you're taking samples from nature, mm-hmm. well, those natural samples, a lot of that biology hasn't evolved to love that tea environment. And so the environment selects in biology. It's almost like basically a law.
Yeah.
The environment selects. And so you created a different environment, and now the biology is working through that environment. And you talk about having the morphology of rods, but I would argue also that there's probably a lot of diversity because it's one morphological group. There's a lot of different species and genus mm-hmm. that represent bacillus rod-shaped. They could be gram-positive, they could be gram-negative. I don't know if you did those gram stainings, so there could be a lot of diversity, but you wiped out the microbes that couldn't persist in that environment. And so now you're just forced to work with the microbes that will work for you in your tea environment, that will work for your farm.
Yeah, just qualitatively under the microscope, you see a massive drop in diversity over time, which just got me thinking about the whole disturbance thing. But one of the things that agronomist Lee Brees, I had him on the podcast, he turned to me and he goes, "Tad, agriculture is not a natural system. It's not natural."
Right.
And I have to remind myself of that, because people always say, go back to nature, do what nature does. Well, nature doesn't do this. It doesn't select for one species or one variety of plant over at the exclusion of all else. And so the amount of diversity in a forest versus what we see on a microbial level in agricultural lands is quite drastically different. And
totally.
I think we have to remember too, like in an agricultural system, every time we harvest a plant, we're removing biomass, we're removing organic matter, we're removing nutrients, and those have to be replaced. And if we can manage that replacement to keep us at a level of stasis that's optimal for, cycle after cycle, that's really what we're going for with soil testing and things like that. So I find it all really interesting and fascinating, but I don't get as worried about disturbance. And some more recent things that have got me thinking about this, and again, I'm talking about indoor beds or pots. I'm not talking about tilling outdoor land. I think there's a lot of reasons not to till, for erosion and other disturbance issues. But if we're talking about in indoor beds, hand tilling or hand disturbing, we know now that heavy metals tend to accumulate in the roots and the stems of the plants more than buds. There's research to support that. So if we can remove those things, we're lowering the amount of heavy metals in our bed, which is a good thing, especially in states where you're regulated. From the presentation and the research by Dr. Punja, we know that this viroid is going to be more accumulative in the roots of the plant. So if we take those roots out, we'll be removing some of that viroid load. So at this point, and maybe someone will convince me of something differently, am inclined to remove as much of that as we can. So I think it's all very fascinating. Do you think we'll ever fully understand all of this because it is so complex?
Listen, I don't think we'll ever fully understand even the human body. And it has orders of magnitude more research funding thrown at it than microbiology, to be fair.
Mm-hmm.
And so I think we'll continue to learn more. And I think 20 years from now, we're going to look back and we're going to say, man, we didn't understand that at all. And just like viruses, we've learned more about viruses and viroids than we ever have before. And we know about this much about them. The techniques and methods and the way they just function. Although, coronavirus has helped with that, with the research and funding to understand that. Mm-hmm. It's like a drop in the ocean on what I think we know about these smaller life forms for a couple different reasons. And it's still pretty nascent, although it's amazing what we've learned over the last, let's say 15 years. I remember the first time I went to a soil ecology conference and they decided to open up the black box of soil, and agriculture just considered soil a black box, just a constant. That's how it treated soil. And it said, we're actually going to look inside and we're going to start understanding the biology. And that was in the 90s. And that's when kind of microbial ecology started, as far as I'm concerned. There was a lot of work done before, but it was very early. And so, yeah, we still have a lot of fun stuff to learn on this.
Yeah, when we talk about the history of agriculture, I think one of the reasons we've been successful is, like you mentioned, that pH is one of the main variables in terms of influencing microbial growth and what microbes are successful. Well, that's where I think the soil testing really was important, because I hear people talk about, like, I'm an organic grower, I don't need a pH anything. And yes, soil has amazing buffering capacities, but this idea that, farmers for years have been targeting, very specific pH ranges, which are not beneficial for plant uptake, but also beneficial for these microbes. I think maybe we were inadvertently getting benefits that we weren't even aware of on a microbial level. And also why I believe soil testing is so important when we're talking about re-amending and managing, a grow.
Totally. You're kind of talking about, I think about confidence, and I think consistency is key. Consistent inputs or consistent practices ultimately yield consistent results. And consistency can be a predictable range. And especially when we're talking about microbiology or organic growing or living beds, it's not going to be consistent. Mm-hmm. But I think that what we want to do ultimately is predict the range of confidence so we know the ups and downs, so to speak, of what to expect from functionality and just all the constituents of the soil. And then, like you're talking about, you can manage that. As long as you can measure it, you can manage it. To think it's static, that's just not realistic in life at all, especially when you're dealing with biology. So that's exactly right.
So rather than what you're saying there is what we're targeting is a range with soils in terms of outcomes.
You have to.
Yeah, it's not.
I think that's the way to manage it. Yeah,
it's not just this finite number. It's a range of numbers, but you can control that. And it doesn't mean that it's bad. We just have to let go of a little bit of our control in these systems.
Yeah, totally. Listen, I used to do a lot of natural studies in grasslands and whatnot, and I used to do annual studies where I would do quarterly and seasonal. I'd go out and take samples and look at the rhizosphere microbes and look at how the population or the community diversity changed. And it changes every season, mm-hmm. depending on the conditions. And a lot of that, although the soil was well buffered as far as pH, the plants had a large influence. And then the temperature had a large influence. And sometimes, the hydrology had an influence there also, where I could see a dramatic shift in microbial community assemblage and populations or concentrations across the growing season, across 12 months or 4 seasons. And then it would come back. And so my predictable range was that annual cycle where I could pretty much predict what the microbes were going to be doing on a seasonal level based on 3 or 4 different parameters.
So you can, if you take a far enough step back, you get that consistency, or you start to see some patterns.
Range.
Yeah,
totally, exactly. Yeah.
So I'm looking at my notes. I know we're running out of time. There's one question I didn't really get to ask you that I wanted to, that kind of fits into what we're talking about here, is from my interview with Dr. Michael Brownbridge, one of the things he mentioned was like, he doesn't understand why there's so many products in some of these, biostimulants or micro products on the market. There's a huge list of microbes of what they're putting in there. Knowing that a lot of these may occupy the same ecological niches or compete for the same space, and like you said, knowing that these microbes are constantly changing in the environment, like, do we want a more targeted approach where we have one microbe, or like mycorrhizal fungus or trichoderma or Bavaria, or do we want a consortium of microbes in our microbial product, or do we want I guess do we need? Is it a waste of money to be buying ones that have all of these in there when we could just be a little more targeted about it, I guess?
Yeah, I'm not so sure that cost is associated with the different concentrations or the different formulas of microbes.
Okay.
I would say that in general, it kind of just depends on the confidence you have in that product and what you think it's going to do. I've seen companies just throw everything in the kitchen sink at a product and just have everything in there because that was their marketing strategy. I'm not so sure how much actual scientific validation they had for the formula rather than just throwing everything in there and saying, hey, everything's in there. Mm-hmm. I think that it does provide for some functional redundancy if you have more species. I am a proponent of a consortia effect, but I would say a responsible or a well thought out consortia effect. That was the thought behind Mammoth microbes or Mammoth P. That's also the thought behind the microbial mass technology. You have microbes, what the microbes are. Your promise is those microbial concentrations and what they're doing for your plant. And I think that's a really good approach. When you start seeing a large assortment of microbes, I really question the consistency. And I would suggest that it's really hard to manufacture with consistency, really large consortia, and maintain that in the bottle over time. I'm not so sure it's bad or good. I would say that it's less defined and more. I would consider them much more general solutions just to help with nutrient cycling. And I would say that more of the functionally targeted consortias are a very interesting approach because there's typically a lot of thought put behind not only the function of the microbe, but the redundancy of the microbe and the environmental resilience of those microbes. And that's why I see in the products that have a limited consortia, there's a purpose for them.
I think that's a good point. And one thing that I think that you and I just kind of take for granted, and that I hope other people will start doing, and many people do, is just set up a trial. Like have a control, test to see if this product in your environment gives you an effect. And you may be surprised by the results. Like I, for example, was surprised by the Mammoth P results we got at Gold Leaf. I did not think we'd see any sort of statistically significant difference in yield, and we did. Because I was like, our soil already has all this, good microbial activity and everything. So that surprised me. And I think that's worth revisiting to see if we're still getting those bumps. I think this is sort of thing is, people have to do this ongoing. Don't just do one trial, but replicate it. And now we're setting up a trial right now with the Miicrobial Mass at Gold Leaf. And I'm excited to, hopefully get to document and share those results. But I think people need to test it out because I I feel pretty confident saying that there's a lot of microbial products on the market that give micro products a bad name, whether they just, took a bunch of powders out of China that were supposedly these concentrations, mixed them all together because they saw a research paper on it that said that they were beneficial. That stuff's out there, and some of them are quite popular. And frankly, I don't think people are getting the results that they should be getting for what they're paying for it. So.
Yeah, totally. I think that's absolutely right. And that, me, that's the approach I've always taken, kind of this scientific approach. I like bringing validated innovation into the market to help the success of growers. That was the whole intention, kind of the purpose I have professionally and as a scientist, to be fair, at the university. And so it's really important to measure those results and to define success, and talking with the manufacturers to understand what those expectations, what is the product intended to do, and then you can make sure you can measure it to get those results. If it increases yield, if it's supposed to increase yield, is there validating data that the manufacturers have conducted studies that shown that it's increased yield? And so you can actually start to have some confidence that you would expect that, so you can measure it. Same thing for a quality metric. I've seen products that, promise quality metrics. Well, if you can measure it and you have validating data that supports that, let's use the product for its intention. A lot of cases, there's not a real purpose. There's not a lot of data-backed products out there. And so there's not a lot of expectations. Just use it and you're going to see good results. Well, okay. But it's really hard to measure that if you can't define it. And so on both sides, I think the manufacturer should set clear expectations of what the product does for growers, and the growers should set real expectations of why they're using the product and be able to measure that. And so yield and quality are pretty simple things. If you think about early stage cloning products or rooting products, let's make sure we can measure root mass and add these products in to see more root growth. And so I still think biology should be targeted to a specific function, a specific outcome. Why am I using this? I want to measure it, and it should do it. Just like nutrients and everything else, water does something, nitrogen does something, phosphorus does these things that we can almost measure. If we can't completely measure, we should treat microbes and microbial innovations the same exact way, with rigor.
I totally agree.
Yeah.
Well, I super appreciate you taking the time to chat with me today, and I look forward to some of the new innovation and things that you have going on, and following up on this trial with the Microbial Mass product. So yeah, thank you.
Yeah, looking forward to it. Great seeing you, and thanks everybody.
That was Colin Bell, and you are listening to the Cannabis Cultivation and Science podcast. I'm your host, Tad Hussey. If you like the podcast, please leave us a rating and review or give us a follow on Instagram. You can also sign up for our newsletter on our website homepage to stay up to date on the latest research and information. Thanks for listening.